This video demonstrates the standard procedure for processing soil samples for microbiome analysis, as well as for bulk soil intended for physical-chemical analysis. It also provides instructions on how to assess soil moisture, determine soil pH, and quantify particle size fractions. Freeze soil samples for microbiome analysis at -80 degrees Celsius as soon as possible after collection. Ensure all bags are clearly labeled and tightly sealed. These will be the samples for shipment. Samples should be shipped within 2 weeks of collection. If immediate processing is not possible, store soil samples at 4 degrees Celsius. Ensure the bags are properly sealed to prevent moisture loss. Samples should be processed within 24 to 48 hours after collection to avoid changes in soil properties. Thoroughly mix the soil collected from the 5 sampling sites within each plot in a clean, dry container to obtain a representative bulk soil sample. To measure soil pH, you will need an analytical balance, a clean spatula or spoon, deionized water, and a calibrated pH meter. Start by transferring 2 grams of well-homogenized soil from the previously prepared mixture into a clean container. Add 20 milliliters of deionized water and mix thoroughly to form a uniform suspension. Measure the pH using a calibrated pH meter and record the value once it stabilizes. To determine soil moisture content, you will need an analytical balance, a 500 ml glass beaker, a clean spatula or spoon, and access to a drying oven. Start by weighting a 500 ml glass beaker to the nearest hundredth and record the weight. Fill the beaker with approximately 200 ml of well-homogenized field moist soil. Weight the beaker with the soil to the nearest hundredth and record the weight. Place the beaker with the soil in a drying oven set at 65 degrees Celsius and dry for 48 hours. Remove the samples from the oven and allow them to cool to the room temperature before reweighting. Finally, record the oven dry weight to the nearest hundredth and use the formula provided in the protocol to calculate soil moisture content. To assess fraction size quantification, you will need an analytical balance, a clean container, and a 2 mm sieve. Pass the dried soil through a 2 mm sieve to assess the proportion of coarse and fine fractions. Weight the coarse and fine soil fractions separately and record the weight of both soil fractions. Sieve at least 400 grams of the homogenized bulk soil sample through a 2 mm mesh to remove coarse particles, stones, and roots. Transfer 250 grams of the fine fraction into a properly labeled zipped plastic bag. Leave the bag open to air dry at room temperature and store it in a clean, dry environment until shipment.